Okay guys, so today we're going to take a look at some of the food samples that you guys consume and take a look to see what type of macromolecules can be found in these foods. So we know that there's 4 major organic macromolecules in our food. They include your carbohydrates, your fats, your proteins, and your nucleic acids, which are basically your DNA and RNA. For today, we're only going to experiment on 2 of the main food groups, which is going to be your carbohydrates. We're going to look for 2 types of carbs, starch, which is a complex carbohydrate, as well as simple sugars, like your glucose and sucrose. And we're also going to take a look at our proteins and see if our sample contains proteins. Now, before we get started, we need to figure out how we actually running this experiment. So for us to be able to understand what each of our food group contains or what each of our food sample contains, we're going to use what we identify as indicators. So right here I have 3 different types of indicators. Indicators, simply put, are going to be the solutions that are going to change color in the presence of a very specific molecule. Otherwise, the color is staying as original color. Let me give you an example. So one of the samples that we have here is Benedict solution, and you can see the color of this indicator is blue color. So the Benedict solution is identifying simple sugars when it's heated. Now, if my sample contains simple sugars, then the color will change from the blue to a range of either yellow, orange, or red, depending on how much sugar we have in the sample. So if I have sugar in the sample, but the sugar amount is little, then your color will be yellowish. If there is more sugar, then it will be orange. And then lastly, if there is a high concentration of simple sugars in it, then your color will be red. Now, if there is no simple sugar in your sample, then the color stays as the original blue color. Second sample we have is ninhydrin. Ninhydrin is also have the clear color. Notice that this sample does not have any basically a foil wrapped around it, and the reason for it is because it does get reactive with the sun and the light, so we are need to protect it. But if I open up the stopper, you guys can see that the solution is almost a clear color. It has a yellow tinge to it, but it's a very clear solution. Now, ninhydrin will be used to identify proteins. So if you're looking at a sample that does contain protein, and we heat that sample with the presence of ninhydrin, then your sample will turn to a purple color. However, if the sample lacks protein, then the color will stay as clear. Last indicator that we have in this case is our Lugol iodine. So this sample is basically, Lugol is basically, or iodine is what you guys use to clean a wound. The color of this, it doesn't look yellow, but because it's super concentrated, but when you add it to the solution and it's diluted, the color is actually yellow. Now, what is Lugol iodine identifies? It is identifying your starch. If there is a starch present in your sample, then the color will change to bluish black. And if there is no starch present in your sample, then the color will stay as the yellow original color. So for us to kind of test this on a kind of a sample, we're going to run this experiment with our 3 indicators. So I'm going to start with our Benedict. So kind of a reminder, guys, Benedict is going to be the one that is identifying simple sugar when it's been heated. So right here in my tubes, I have 3 samples. The first one is a sample of a protein or a solution. The second one is a sample of a starch, and the third one is a sample of a sucrose or simple sugar solution. So again, the only time I will expect to see a color change is when I'm using Benedict in combination with my simple sugar solution, and that's where I will see a color change of either yellow, orange, or red, and the other 2 samples should stay as blue regardless of how much heat I apply. So here we go. So I'm gonna start my sample. So I'm gonna add a few drops of my Benedict solution to each of these. And again, remember, what you wanna do is mix your sample, mix, and then I'm gonna place them inside my water bath. Now, if I do start to see color change, then I'm going to assume that the glucose or simple sugar was present. If not, I'm going to continue heating it for 5 minutes until the time runs out, and then I'm going to say that the sample was negative. Now, while we're waiting for this, I'm going to run the second test, which is the Lugol iodine. Again, Lugol iodine will be used to identify starch. So, same sample that we did here, we're going to repeat it. We have a solution of a protein, we have a solution of a starch, and a solution of simple sugar. So what I expect to see, if my prediction is correct, the one that has the protein and simple sugar will stay as yellow because the indicator Lugol does not identify anything. However, when I add it to my starch tube, the Lugol iodine would interact with this and would, as a result, will be identified as. So let's go ahead and add our sample to our Lugol iodine to our 3 sample. And again, we're looking for yellow if there is no reaction, and we're looking for bluish black if there is a reaction. So here is a few drops. And again, so let's take a look. Yellow means no reaction. This is our simple sugar tube. So technically, we will not be expecting to see any color change. Second, look, that was our starch sample. When I added the Lugol solution, you can see it changed to that bluish black. Third sample is our protein sample. And again, notice that the protein also stayed at that yellow color without having any color change, indicating that our protein sample has no starch present in it. So here we go. So this is our sample right here. So let's go ahead and take a look at our next sample, which is going to be ninhydrin. So I'm going to turn this around, and we're going to take a look at our sample. And add our ninhydrin to our 3 sample. Again, we have a protein sample, we have a starch sample, and a simple sugar sample. So I'm going to add a few drops of this, a few drops here, and a few drops here. Please do note that ninhydrin is another sample that needs to be heated for us to see the reaction. So I'm going to mix these samples, again take them out. Look, remember we're looking for a purple color. So I'm going to take my original samples out for the sugar and put these ones in for them. And don't do what I'm doing because your tubes might be hot when you're doing this. So I have taken out my sugar sample or simple sugar test, and I'm placing my protein test back into the heat. So let's take a look at this. The first sample we have right here is was our simple sugar sample. You can see a very beautiful orange color, which indicates that this sample is positive for simple sugar when a Benedict was added. The next sample we have is a starch. So a starch is, again, we started with a bluish color when we added our indicator, and after heating you notice there is no color change again here. And this is our protein sample. Again, you notice that there is no color change even looking at your protein sample. So basically, while we're waiting for this to heat up, I want to just kind of go over this with you guys one more time. The 2 samples that you will be heating will be your sample for the Benedict solution as our indicator, and the second sample that you guys are going to be heating is your ninhydrin sample. A starch sample does not need to be heated. If the starch is present, as soon as Lugol iodine reaches it, you will expect to see a color change. So we're going to leave these samples be for a few minutes. While this is ongoing, I want to show you the setup that you guys are going to basically collect data on for the experiment today. So right here, what I have is 4 samples of food, and we are going to test to see what each of these food samples contain. So right here I have a granola bar. This is some oats. I have some eggs, and then in the last one I have bread. So understand when we're running this test, guys, that you want to test for all of the 3 samples, right? Or all of the 3 indicators. For instance, let's say take eggs. So eggs, I'm going to have a tube that is testing for protein. For egg, I have a test tube that is testing for your presence of simple sugar, and I will also have another tube for my egg that is going to be testing for the starch. And I'm going to repeat this sample for here. So if you take a minute and I say, okay, how many test tubes total I need to run this experiment right here with these 4 samples, you will need a total of 12 test tubes, right? 3 for each sample to test each of the indicators. Now I'm gonna shift because I want to finish up the setup for you guys here. So right here we have our sample again. I don't know how clearly you will see this, but notice the first 2 tubes have no color change, so they're staying the original color, but the last one has a purple tinge to it. So this purple tinge basically identifies that your sample has proteins in it, versus when you compare it to the other 2 samples, the samples are clear, which means no protein was present in it. And you always want to use this as an indicator for your experiment to see if something was tested positive or negative. So I'm going to show you how my typical setup is for one, and then I'm going to come back and show you what the result of each of these experiments was. So let me show you one setup first. So here I'm grabbing 3 test tubes. So I'm going to use my wax pencil and I'm going to label each of these. So the one that is going to be testing for simple sugar, I'm going to label that with B for Benedict. The one that is going to be testing for protein, I'm going to label it with N for anhydride. And the last one, which is identifying the starch, will be using glucose, so I'm going to identify that with L. Now again, we're testing each sample for the 3 possibilities. I'm going to start with my granola bar. So I'm going to grind this. And the smaller your food sample is, the better it would be for you to run your experiment. So this was easy. So I'm going to use my spoon to add a little bit of food sample to each of my test tubes. And again, each food sample will be tested for simple sugar, will be tested for a starch, and will be tested for proteins. So here's my 3 food samples. Now, for this, before I add my solutions, I do need to add a little bit of water. The water is not essential, guys. It's just for something to kind of wet your sample and make it easier for you to mix your sample with your solution. So again, the amount of water is not as essential for you to know. So here I'm going to mix my sample, make sure that it's properly mixed. Again, to the one that is labeled B, I'm going to add my Benedict to see if there is simple sugar present. To the second one that is N, that's testing for protein, so I'm going to add my anhydride. And to the third sample, which is my Lugol iodine, I'm going to add my Lugol iodine to test to see if there is any starch present in it. Again, the one that is for Benedict and anhydride needs to be heated, so I'm taking these samples, mixing them and placing them in water. Notice that this sample, which is my Lugol iodine, already has changed color to bluish black, which means that this sample is testing positive for starch. So that's a really good indicator. Reminder, guys, this sample for Lugol iodine does not need to be heated. So we're gonna watch these samples for any color change, and when that happens, I will let you guys know. Now, please do know that I'm not gonna show you every sample that I have here. I will show you your final results. So what I'm gonna do is I'm gonna do my samples, heat them up if they need to be heated, and I'll show you, I'll come back with the final result for your data, okay? Okay guys, so I've run the experiment, and remember that I was supposed to heat Benedict samples as well as your anhydride samples. So I've done that. I've run all of the tests for all of our 4 samples. So what I'm going to show you is samples one at a time and tell you what color changes I saw. And you guys can also observe it right in front of you. So you should have your lab activity in front of you and write the name of the food sample. And on the first table that you have, you should be able to identify, you should be able to write down the colors that we have here, and then I'll talk about the second table later on. So what I'm going to start with is our first food sample we have right here, which is our egg sample. So again, the first tube right here I have is for the Benedict sample, which is again testing for simple sugar. Notice for the egg, the sample of the Benedict stayed blue color, which means there was no reaction. Second tube for the egg, we tested for proteins, and as you can see, the color has turned purple. Third tube tests for starch using your Lugol iodine, and you can see the color is yellow. So that's your first food sample. For the second food sample, we have the granola bar. So this sample is testing for your Benedict solution. So the color you're observing is orange. The second sample you have is testing for anhydride, and you can see there is no color change. So you still see the clear sample of water right here, no change in color. And third one is the starch sample, and you can see the blackish color of the starch. Oh, sorry, I should say the blackish color of the Lugol iodine. So this was our granola bar. The next sample we have right here is our oats. So the first one we have is Benedict solution. Again, notice that the color is recorded as blue. If I look closely, it has a really kind of light greenish tinge to it, so you may also record it as green if you choose to. This is our Lugol iodine sample. The color is black. And this is our anhydride sample. The color is purple. So this is our sample for the oat. And the last sample we have here is for our bread sample. The first tube you can see here is for our anhydride test. You can see there is no color change, so definitely you can label it as clear. The second tube we have is, you can see the sample completely turned black. So the color for this one would be black for your for your Lugol iodine sample. And then the third one, which is for your Benedict solution, after heating it has turned to orange color. So these are our 4 food samples that we have tested. So based on the results you guys now have, you guys are going to complete your second table. The second table is asking you to basically put a plus or minus for the tested that are positive. Put positive, of course, plus for the positive, and if the indicator did not change color, that means that was a negative test. So to just kind of give you a hint as far as how you do this, going back to our first sample for the egg, right here you recorded blue color on the first table for the Benedict. So when I go to the second table, I would write a negative for this one because the color change was absent. For the protein sample, I recorded the purple color, so I would put a plus sign because that's a color change indicating my sample was positive for proteins. And third, we have the starch sample, and you can see there is no color change in this case, and therefore for your egg sample you will record negative for your complex carbohydrate or starch. Now you guys are going to repeat this for the other 3 samples, and after you've done that, you guys are going to complete your questionnaires at the end of the lab activity. Okay, so that's it for us today.